What are some of the most common misconceptions about CRISPR engineered stem cell therapies?
Yeah, I think there's yeah, it's a really good question. I mean, I think number one potential misconception is that they could be dangerous. Yeah, because you are manipulating the gene of the stem cell, and stem cells are long-lived cells that give rise to many other cell types. So a stem cell will develop into a neutrophil. It'll develop into a red blood cell. So it gives rise to many different types of cells. So if you're making a gene edit in a stem cell, that gene edit could be permanent and it could potentially be detrimental. And I think one misconception about gene edited stem cell therapies is that they could be dangerous. Now we have data in at least a dozen patients or so where that's suggesting that it's not the case. It's still early days, but I think we've to a certain extent de-risked the safety aspect. So I think they could be potentially safe. Obviously, with clinical development, you never really know these things until you've treated, hundreds, if not thousands of patients. I think the other misconception out there is that, it gets back to an earlier point. CRISPR has democratized the concept of gene editing. So conceptually, it's very easy to use CRISPR to edit a cell, to edit, mice, to edit, small animals, the cells of small animals. So people, I think, make the incorrect assumption that because it's easy to use CRISPR, that it's very easy to apply that technology to clinical outcomes. And I think, that's a very dangerous assumption to make because, there's still a lot of preclinical safety work that one needs to do before they get into the clinic. And the FDA and the EMA and other health agencies are still learning about these technologies as well. So, these things are easy to use in the context of the lab, but they're a bit more challenging to translate into the clinic, and for good reason, because it's a new tool, it's a new technology. We don't want to put patients at risk. And so, just because it's easy to use in the lab doesn't mean that it's easy to translate into the clinic. And I think that's an important thing that, we need to emphasize. And it's a misconception.